everyone, welcome to this informational video on a novel probe discovered for non-invasive brain imaging, brought to you by the Demystifying Medicine program. Before we dive into this new discovery, let's review the basics. To start, what is brain imaging? Brain imaging involves the use of different techniques to non-invasively visualize and study the structure and function of the brain. In the past, non-invasive brain imaging did not exist. Instead, to understand the human brain, scientists had to use more invasive methods such as dissecting and comparing the postmortem brains of healthy and unhealthy individuals. However, now scientists can see inside an individual's brain in real time without having to cut it open. There are many types of imaging techniques, but today we'll be exploring a technique known as fluorescence brain imaging. So, what is fluorescence brain imaging? Fluorescence brain imaging is a technique used to help see structures and processes in the brain. As indicated, it is based on fluorescence, which is the ability of an object or substance to emit light after absorbing some type of light or electromagnetic radiation. A common example of fluorescence would be the glowing fish seen in aquariums. It is usually based on the visible region ranging from 400 to 700 nanometers in wavelength. Fluorescent probes, which are compounds that emit fluorescent light after excitation, are injected into the bloodstream. The light emitted from the probes contrasts with the brain tissue and is used to visualize images. A strength of fluorescence brain imaging is its high spatial and temporal resolution. This means that more detail can be collected on where and when areas of the brain are active. However, a limitation is that deeper tissues and organs in the body require light of longer wavelengths to detect fluorescent probes. Also, the blood-brain barrier adds a challenge. The blood-brain barrier consists of a highly selective border of specific cells found around the blood vessels in the brain. It strictly controls which substances are allowed to move from the bloodstream to the brain tissue. There are many benefits to using a probe that can go past the blood-brain barrier. However, majority of current probes are unable to do so. This is where the novel fluorescent probe stands out. Developed by chemists at Rice University and Stanford University, this probe is the first of its kind that is able to travel across the blood-brain barrier. So let's get into how this exciting new molecule was created. From previous research, the chemists decided to focus on using the second near-infrared spectrum, aka NIR2, ranging from 1000 to 1700 nanometers in wavelength. NIR2 probes have been found to have higher resolutions at deeper penetrations than NIR1 or visible probes. With the goal of developing a small NIR2 fluorescent probe, they had to keep in mind various factors such as stability, molecular size, signal strength, molecular weight, and more. In the past, specific boron difluoride aka BF2 molecules were used as probes. From research, a new group of BF2 formazonate molecules was discovered with more useful properties. The term moiety refers to a specific part of a molecule, such as a functional group. When an aniline moiety was slightly altered with different electron-donating groups in the BF2 formazonate molecule, the properties of the resulting molecule could be controlled. Thus, by modifying the aniline moiety in BF2 formazonate probes using complicated calculations and research, they were able to develop 8 NIR2 fluorescent probes with small molecular weights, high brightness, high stability, and the capability of crossing the blood-brain barrier. From this, they tested the probe's ability in mice to tell the difference between normal brain tissue and glioblastoma, which is an aggressive brain tumor. First, they applied the probes in vitro to glioblastoma cells in culture. The glioblastoma cells were incubated in small concentrations of the 8 probes labeled as BF1 to BF8 for 20 minutes, which was followed by washing and NIR2 fluorescence imaging. The results showed that the probes had bright NIR2 fluorescence. Also, the fluorescence of the new probes remained strong for a long time. In contrast, the fluorescence of indocyanine green, aka ICG, which is the first fluorescent probe in the NIR region approved by the FDA for in vivo use, decreased significantly over 12 seconds. Overall, these findings show that the novel probes had brighter fluorescence and longer stability than ICG. When evaluating the properties of the new probes, they found the probes to have significant potential for crossing the blood-brain barrier. Upon further testing and calculations of diffusion, they found that the BF1 and BF6 probes displayed much higher blood-brain barrier permeability than the other new probes. Overall, this finding showed that out of the 8 probes, BF1 and BF6 had the most potential for crossing the blood-brain barrier and being used for diagnostic imaging. From this, the probes were tested in vivo in mice. The probes were injected into the mice via a vein in their tails. This was followed by NIR2 fluorescence imaging. For comparison, mice were also injected with ICG. The results showed strong BF1 and BF6 fluorescence signals with high detail. In addition, as seen from the images, the ICG fluorescence was mainly seen in the blood vessels instead of the brain tissue, since ICG cannot cross the blood-brain barrier, but the new probe signals are seen throughout the brain. Overall, these findings show that the new probes displayed stronger signals and the ability to cross the blood-brain barrier when compared to ICG. Finally, the probe's effectiveness for imaging glioblastoma was tested. To review, glioblastoma is an aggressive brain tumor, and its growth is dependent on the formation of new blood vessels, which are usually abnormal. First, glioblastoma cells were introduced into the brains of the mice and left to develop for 3 to 4 weeks. Then, ICG was injected into the mice like before. This was followed by NIR2 fluorescence imaging. After 24 hours had passed, either BF1 or BF6 probes were injected into the same mice, followed by imaging. The results showed that ICG was able to display the abnormal blood vessels but was unable to cross the blood-brain barrier. With BF1 and BF6 probes, uneven fluorescence intensity was found throughout the brain. After this, the mice were euthanized to study their brains ex vivo. When comparing the in vivo and ex vivo data, they found that normal brain tissue showed higher fluorescence intensity than glioblastoma. Lastly, they found that compared to ICG, the BF1 and BF6 probes were able to better differentiate between normal brain tissue and glioblastoma. Overall, these findings showed that the novel probes have greater effectiveness for imaging glioblastoma than ICG. A limitation of the study is that the probes have only been tested in mouse models. A future step would be to research and test whether these probes could be used in humans as an imaging tool. Also, the preliminary study found insignificant toxicity in the major organs of the mice that were injected with the probes. However, further studies must be done to see whether the probes are safe for use in humans. Overall, the chemists created several amazing probes that have the potential to be very beneficial in the field of medicine when compared to current probes. For example, they could be used to diagnose glioblastoma. They can also be used to help see areas that are deeper in the brain. Finally, they could be used in image-guided surgery to visualize the border between normal brain tissue and tumor tissue. This concludes our short video on the novel probe discovered for non-invasive brain imaging. It really is possible to see beyond the blood-brain barrier.
Created using Powtoon.